Hi, my name is Blaze, and welcome to my playlist on modeling gene regulation networks. The goal of this playlist is to show various Python modeling techniques and apply these to biological systems. This first video has an objective of a big picture introduction to gene regulation. What are some state-of-the-art concepts? What are some basic concepts? And what is a gene regulation network? Are questions that we will attempt to answer today. So the first thing we need to understand is, how do cells survive? There are many potential answers to this, but the one we will focus on is they can regulate themselves and thus respond to environmental conditions, environmental changes, etc., etc. For example, they can control the rate of their own metabolism. But understanding this regulation network, a gene regulation network within a cell, will lead us to the ability of developing medicinal techniques, improving cell modeling, and more. Today I will focus on prokaryotic cells, specifically E. coli, as this will provide us with a basic understanding of gene regulation networks and where the field is right now. Eukaryotic cells, such as yeast and other mammalian cell types, maybe have a lot more complex gene regulation networks, which I will talk about in future videos. But essentially, if this is an E. coli cell, we have DNA in the middle, we have a plasma membrane along the outside. E. coli have this flagellum tail on the end. We have these ribosomes. Ribosomes conduct our translation. We have this cell wall and other components, but this is basically what we need to understand for the purpose of this PowerPoint. Now, the central dogma of molecular biology, so it's termed, is DNA turned into RNA, mRNA specifically. There are many types of RNA. I will not talk about any others except for mRNA. And that mRNA will turn into protein. Now, this protein then needs to get some post-translational modification to turn into an active protein that has a function. But the idea of the central dogma of molecular biology is we have DNA that stores long-term or information for the long term. You have mRNA that stores information for the short term. And then you have proteins, active proteins, which are the effectors of the cell. Now, you can have some RNA effectors, but specifically mRNA doesn't usually have an effect. And if you have more questions on this, I would refer you to other biological videos on YouTube or Wikipedia or things like that. I just want to gloss over this and get to the modeling aspect of it. But an important idea to understand when it comes to the central dogma of molecular biology is, as scientists, as engineers, as pursuers of controlling gene expression, we can insert ourselves in any step of this process to control active protein output. For example, if I were to say repress transcription of DNA to RNA, we would not get that protein at the end. Or if I were to repress translation, I would not get this protein. Or if I repressed post-translational modification, I would not get that active protein. So there are many ways that we can control gene expression. This is the transcriptional regulatory network in E. coli. You can see it's really complex. So what each node is essentially a gene, and then each line connects that gene to another gene, so it has some control of another node, and then that node has more control on the another node, etc., etc., etc. So it's really complicated, and this is just transcription regulatory network, or the mapping of a transcriptional regulatory network. The translational, the post-translational modifications of a protein or a peptide this are not included in here. And so this is just what specific genes affect on each other. But of course, if we go back to our central dogma of molecular biology, this active protein may repress translation. It may modify our mRNA. There's a lot of different levels when it comes to expressing and the activators or the effectors of the cell. And so this helps us understand that cells are really complicated, and modeling them and building a cell in a lab with as complicated as a network as even E. coli, a very small prokaryotic cell, is very challenging. This is a state-of-the-art synthetic network that was created in 2016. Essentially, here you have 42 parts that there's some auto-regulation, there's some upstream regulation, there's some downstream regulation. There might not be downstream, no, just upstream. That all of a sudden control the expression of this red gene that then fluoresces yellow. YFP is yellow fluorescent protein. But the idea of this being that this is 42 parts to control the output of one. But you can understand here that one of these little nodes is going to have a lot more parts contributing to that expression of that specific gene, and then that gene is going to control a lot more, and so a lot more genes and a lot more cellular activity than even the most state-of-the-art methods that we have created in the lab. And so obviously we still need to increase complexity if we want to build synthetic cells that are even as complicated as E. coli. One of the ways we use to model gene activation and repression, this is just the activation function, is what we call the Hill activation function. And if you've ever ridden in a taxi or something like that uses a stepwise function, so in a taxi, if you drive, say, a half a mile, your fare will go up, it'll jump up to the next price, and then it'll jump up again. We can't do that in cells. We can't flip a switch. We can't turn the light on with just a simple switch. Instead, we have to model analog functions, because biology is inherently analog and concentration dependent. And so if I have this simple network of X promoting Y, or X, the concentration of X influencing the Y promoter activity, which is essentially the controlling the activity or the expression of the Y protein in this case, you'll see that with small amounts of X, you're not going to get a lot of Y. But then when you have more and more X, you get higher and higher amounts of Y. And this is the function we use to model this. I will probably get into more detail with this and how this works later, but again, this is just an introductory video. And this is a very simple network, just X activating Y. We can have X repressing Y, which is the reverse step function, or probably be like that. And I would refer you to this website. I'll link it in the description. So when you get beyond those simple regulatory networks, you can see how maybe a Hill function would come into play with more than one input or more than one protein input. Here we just worried about the X input affecting Y. But here, if you have two inputs, say X and Y affecting Z, you're going to have to model that a little bit differently. And so it quickly gets more complicated. This is from a 2003 paper that models AND gates, NAND gates, and I think they also did an OR gate of some sort. But they showed you how complex it gets even just with having two inputs. And so if you have 42 inputs, you're not even going to be able to visualize this on a graph. You're just going to have to track the concentration on a graph by itself. But essentially, all of this can be defined with Boolean logic to some extent. Boolean logic meaning like truth tables, and even though that it's not going to be flicking the switch on or off because biology is analog, we can model it in that way. Anyway, I hope this was a good introduction to what I will be talking about in this playlist. I will follow this up with more in-depth definitions, more in-depth modeling, and perhaps some Python techniques. I don't really know where this is going to go. However, I do want it to be useful to the viewer in terms of being able to apply these concepts into Python for their own modeling purposes.